Hello, Lakshmi. Hello, Lakshmi. How are you? Okay. Am I audible and visible? So today I'm going to discuss about mainly plant vector. Okay, Agrobacterium tumefaciens. This is bacteria, and it is known as the natural genetic engineer. Hello. Okay. Just a minute. Okay. See. Uh, So agrobacterium tumefaciens is the natural genetic engineer. Okay. So let's talk about this bacteria, how it is useful. Okay. Already I have discussed this thing, right, Lakshmi? Transfer DNA, TI plasmid. Just I inform that TI plasmid is utilized in case of plant gene cloning system. है कि नहीं? During the first class of the gene cloning, I have discussed about TI plasmid. But shortly I have discussed. Today I'm going to discuss elaborately. Okay, just a minute. Give me one separate color. Okay. Now see what is happening here. If you consider about the TI plasmid, okay, TI plasmid which is present in which bacteria? Agrobacterium tumefaciens. Okay, TI plasmid present. In TI plasmid, okay, few regions are there, important regions. First of all, you have to know, first of all, that is the virulence region. So, due to the presence of this region, virulence is there for this plasmid. It can infect to another system. Therefore, it is known as the virulence region. Okay, this portion is virulence region. Now, in this system, another thing is left border should be there, right border should be there for which region? tDNA region. So, important part of this TI plasmid is actually tDNA region. In tDNA region, first of all, left border, then few plant hormones are there, auxin, cytokine, and opine. These three regions are there. Okay, auxin, cytokine, plant hormones. These are auxin and cytokine. Right, and after opine, what is present? Right border. Okay, so tDNA region means left border, auxin, cytokine, opine, and right border. Okay, so due to this tDNA region, whenever this TI plasmid is entering into any plant, what is happened now? Some tumor structure is developed. Crown gall tumors are developed. Why? Because in plant, these two hormones are there, auxin and cytokine, but this bacteria containing this TI plasmid, what is happening? They are also producing auxin and cytokine. So there will be overproduction of these two growth hormones. Due to overproduction of these two growth hormones, what will happen? There will be tumor formation. Okay, tumor formation in the leaf, in the plant. That is known as crown gall tumors. Okay, and crown gall tumors are actually due to what? Agrobacterium tumefaciens. Whereas if you consider another strain, that is Agrobacterium rhizogenes, it is actually infection will be like exactly same way like TI plasmid here, but the name of the disease that is known as the hairy root disease in plant. Okay, crown gall tumors. Another one is hair root disease. Clear? Now, opine here, opines means mainly those are amino acid derivatives, those are utilized by the bacterium. Okay, but auxin, cytokine, those are plant hormones. Okay, now see here, same thing even. The synthesis of the plant hormones, auxin and cytokine by enzymes encoded in the tDNA enables the plant cell to overgrow, thus forming crown gall tumors typically induced by Agrobacterium tumefaciens. So, crown gall tumors is actually produced by tumefaciens, whereas rhizogenes is producing hair root disease. Okay, clear? But it's the same thing. TI plasmid is present. Okay. Now, in this case, what is happening? What are the different components of tDNA? As I told you, if you consider this is the tDNA, okay. In this case, if you consider what is present, first of all, what is present? This is the infection gene. That is the virulent gene. Okay, vir gene. Okay. So virulence region. So this is virulence region. I'm writing just V. Okay. Now after that there will be one border, left border. Okay. After left border, what is present? What is present? Auxin present. है ना? So I'm writing it as the A. So if you consider A present, one region is there, that is A. Then second region is cytokine. Then another is OP, O. And now after that, which region is coming? Right border. Okay, up to this, these are things. So now gene. I told you auxin, cytokine, opin, these are the gene products. Okay, but there should be some name of the gene which are actually expressing these proteins or hormones. Okay, so here for the auxin, this is actually IAAM. Auxin biosynthesis is happening encodes enzymes tryptophan 2 monooxygenase. You don't have to remember. If possible, you just remember the name of the gene. OXL TNSI, OX2 TMS2. These two actually involved in the auxin biosynthesis. Okay, these two. Now, if you consider about the IPT, TMR or cytokine. So in that case, cytokine synthesis is happening from these genes, IPT. No pun in biosynthesis, we are not considering this. Now 24 base pair left and right border sequences. So left and right border, I told you na, left border, this part and this part. These are actually mainly 24 base pair sequence. Since tDNA is bordered by 25 base pair repeats. So left border, right border means 24 base pair repeat. Okay. Now going to other genes. This you have to remember. This is very important. I am writing VI. This table is very important, means in this TI plasmid, one region I told you, that is virulence region. Okay. In virulence region, only one gene is not present. Several vir genes are present. Okay. Vir genes means vir A, B, C, D. D may be one, D1, D2. Now E1, E2, vir G, B, B already I told you. So A, B, C, D, E, G. So different types of vir genes are there which are participating in different processes. Okay, so here in this position, vir A से लेकर from A to which gene? E gene. Different genes will do the different work. See, vir A means It is encoding a receptor for acetylsyringone. This is actually phenolic compound. Okay, so here it is actually producing a receptor. Receptor for what? Receptor for acetylsyringone that functions as an autokinase, also phosphorylates VG protein constitutive expression. That means in this case, what is happening? Whenever, if you consider key, this is one plant cell, okay. Now, whenever there is any wound is created in the plant cell, what will happen? There will be secretion of lot of amount of phenolics and few sugar molecules. Those phenolics will actually give the signal to VA. Okay, so these molecules actually specially known as the acetylsyringone. That is actually phenolic compound. Okay, this molecule it will bind with the VIR receptor. VIR A is actually giving the receptor, expressing one receptor protein. Whenever it is binding with the receptor, this VIR A actually activated. Now VIR A activated, it will activate VIR G protein. Okay. Now what is the purpose of doing the VIR G activation? G will bind a DNA binding protein, probably forms dimer after phosphorylation by VIR-A and induces the expression of all VIR operons. So first of all, expression is done or activation is done VIR-A, then VIR-G, and it is actually DNA binding protein, and other functions will do different VIR-G genes. How it is happening from another picture, it will be clear. But after the discussion of those pictured or schematic diagrams, you have to come back to this table, because in this table, what are the functions of all the VIR genes is properly written. Okay, just I'm reading it. I know ki after reading you can't understand what is going on, but I'm reading it. Okay, see, where B is membrane proteins possibly form a channel for tDNA transport. So in this case, it is making one transport. Where C means it has the helicase activity. It binds to the overdrive region just outside the right border. So it will bind at the right border, involved in the unwinding of tDNA. VD is actually D1 has topoisomerase activity. So unwinding the DNA, it binds to the right border, not the left border. Here, right border of tDNA and prevent attack by exonuclease. So VD is actually binding with the DNA in order to control the exonuclease activity, in order to prevent the exonuclease activity, and it is binding at the 5' region. And VD2 is an endonuclease. This is endonuclease activity, and it is cutting the phosphodiester bond. So in this case, what will happen? Finally, this tDNA, so left border, right border, tDNA, after that, tDNA will be removed. Okay, so whenever plant would there will be wound in the plant, secretion of polyphenolic compounds will be there. Those polyphenolic compounds will give you the signal to the plasmid. Okay, now VIR genes are responsible for the cutting of all the VIR genes actually involved for the cutting of this tDNA. tDNA means auxin is present, cytokine gene present, and open gene present. So together, these things actually form the TI plasmid. And this part, this DNA, this fragmented DNA, it will go inside the plant cell. Okay, not a whole TI plasmid is entering into the plant cell. Okay, only this plasmid will go into the plant cell. Okay, and this DNA will integrate it into the DNA of plant cell. Okay, So whenever it is integrated, it will overexpression will be there, auxin and cytokine, and there will be tumor formation. Okay. So in this case, this cutting and sending to the plant cell, it is done by the virgins. Okay. So slowly you will understand what is happening. See what this is the plant transformation process. In this case, if you consider discs removed from the tobacco plant, mainly tobacco plants are used for the cloning purpose. And now leaf discs incubated with agrobacterium for 24 hours. So some discs are taken from the this leaf, and it is incubated in the tobacco agrobacterium containing medium. This is actually genetically engineered. So in this case, how it is utilized actually, this process is naturally happening. Okay, agrobacterium is infecting naturally to any plant, and tumor formation is done. This process is utilized during the genetic engineering or recombinant DNA technology. That means through this DNA, this part, you can insert auxin, and at the position of auxin and cytokine, you can add your gene of interest. Okay, and clone it. Cloning you can do. After the cloning, you can insert this part into the plant cell, so that plant cell will be in the plant cell. You will get the engineered gene. Okay. So here also same thing is given. See, discs are taken, paper leaves are taken, leaf discs incubated with genetically engineered agrobacteria for 24 hours. Genetically engineered. That means at the section, tDNA section, some gene is inserted. Okay, now selection media only allows plant cells that have acquired DNA from the bacteria to proliferate. So in the tDNA section, not only gene of interest is taken, along with that, one segment will be there so that you can identify that whether recombination is done or not. Okay, so in that case, marker gene, or you can consider antibiotic resistance gene is also attached with the gene of interest. Okay, so if you grow this cell in the antibiotic, what will happen? Only the recombinant one will grow properly, because the antibiotic resistant gene is attached with it, and other cells will die. So in this way, you can grow it, and you can get the genetically engineered plant. Okay, now see steps of agrobacterium plant cell integration. So this thing I have discussed, this is actually how to what agrobacterium is utilized for the genetic engineering purpose. Okay, now I am going to discuss again the normal case agrobacterium, how agrobacterium is infecting plant cell, and how agrobacterium is creating tumor in the plant cell. Okay, so let's see. First of all, plant stress condition. Stress condition may be, as I told you, wound. There may be some wound, okay, in the plant. So in that case, phenolic production will be there. I told you several phenolic compounds are produced. Specifically, which phenolic? It is given now. It was aceto acetosyringone. Okay, acetosyringone is the phenolic compound released from the plant. Due to that phenolic compound, it is giving actually signal to bacteria. Bacteria means agrobacterium. Inside the agrobacterium, Ti plasmid is there. है कि नहीं? Now signal to bacteria. Now virulence system activation. Now virulence system is activated. Generation of tDNA complex. tDNA complex is mainly T bacteria is there. tDNA is there. It is actually degraded. Okay, separated from the Ti. Now tDNA transfer. So what will I told you na ki this part tDNA part is actually transferred to the plant cell. So this will go inside the plant cell. the DNA transfer, that is nuclear import, and tDNA integration in plant genome. So what will happen? After that, there will be integration in the plant genome. This is the actually natural. In the natural case, it is happening. Okay. So wound, phenolic compound production, signal to bacteria, virulence system activated, separation of the tDNA. tDNA is transferred from the bacteria to plant, and inside the plant it is integrated into the genomic DNA, plant genome. Is it clear, Lakshmi? Lakshmi, Lakshmi, are you audible? Am I audible? Okay. So your voice is not coming. What happened? It's not working. Let's see. Hello.
Yeah, it got
Ah.
disconnected the first.
Okay. 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 So this is the actual case. A normal case, whenever agrobacterium is infecting any plant, these are the consequences happening. Okay. Now, same steps in this picture it is given. Same steps. Okay. So let's see one by one. First step is the wounded plant cell products produces acetylsyringone. This is the phenolic compound. Now, this is the structure of acetylsyringone. Okay, if possible, and remember, but you just understand phenolic means there should be this type of benzene ring. Okay, with benzene rings there should be OH group.
Wash.
है ना? OH group is there, especially phenol group is there. Along with that, few other things may be possible. Okay. So this is one type of phenolic, that is acetosyringone. So this is released from the plant cell. Okay. Now acetosyringone activates virulence genes. Virulence genes means vir, or the vir genes. Okay. A to E. Now third step is vir proteins synthesize single stranded DNA. Single stranded DNA. So in this case, I have not informed you that during this cutting, only single stranded DNA is entering into the plant cell, not the double stranded DNA. Okay. So in this case, see here, tDNA, single stranded DNA is produced. Okay. Now in the fourth step, tDNA transfer is happening. So tDNA is transferred from the bacteria to the plant cell. Okay. In the fifth step, देखो tDNA complex import to nucleus and integration. So this tDNA is actually attached with the genome. Okay. Now, and tDNA integrated at random site in host chromosomes. A single strand is binding, then there will be replication with the formation of double strand. Okay. And what will happen? Finally, this gene will be expressed inside the plant cell. That means overproduction of auxin, okay, cytokine synthesis, and synthesis of opins. Whenever opins is produced, that opins actually utilized by the bacteria, because I told you nitrogen, nitrogen source. Opin is the nitrogen source for bacteria. So opin, it is utilized by the agrobacterium. So you can consider that opin is again accelerating the growth of the agrobacterium. Okay, simultaneously what will happen? Again tDNA is produced, and it will be inserted into the genome of plant. So this is the main thing happening during the infection by agrobacterium. Okay. So agrobacterium tDNA transfer as a natural case. So this is naturally it is happening. We have used this natural thing, and always we human being always finding uh, what will be helpful for them. Okay. So in that case, this process is utilized for the genetic engineering. Okay. Acha. Here, what is given? Wounded plants secrete sap with acidic pH and a high content of various phenolic compounds, lignin, flavonoid, precursors serving as chemical attractants and stimulates Vir gene expression. Acha. Now, among these phenolic compounds, acetylsyringone is the most effective. And in this case, I told you sugar is also responsible for the signal. Sugars like glucose and galactose also stimulate virgin expression. Okay, acetylsyringone as well as sugar, glucose, galactose. They are also stimulating virgins. Vir A. Mainly first of all activation is Vir A. It is binding with the phenolic compound. Okay. Low open levels further enhance Vir gene expression in the presence of acetylsyringone. These compounds stimulate the autophosphorylation of a transmembrane receptor kinase VIR-A. So in this case, it is actually, oh, I have not discussed this. A few cases phosphorylation may create activation of the enzyme or deactivation of the enzyme. Okay, most of the cases you will see whenever a phosphorylation is happening, that enzyme is activated after phosphorylation. So in this case also that is happening. These compounds, that means whenever phenolics and sugars are binding with the VIR-A gene, those are actually stimulating the autophosphorylation of transmembrane receptor kinase, that is VIR-A. So here starting point is from the VIR-A. That means these compounds means these sugars and acetylsyringone. So acetylsyringone, glucose, galactose, those are actually stimulating autophosphorylation of transmembrane receptor kinase VIR-A. Okay. And at the which position? Histidine residue. Generally phosphorylation is happening, histidine, serine residue. Now it in turn transfers its phosphate group to the aspartic acid residues of cytoplasmic Vir-G protein. Okay. So Vir-A is actually present in the membrane. Vir-G is present in the cytoplasm. Cytoplasm of what? Cytoplasm of bacteria. Okay. So A is activated, then G is activated. Okay, by the phosphorylation process. Now, how production of T strand, how it is produced or separated from the Ti plasmid is given. See how it is happening. Every induced agrobacterial cell produces one T strand. Okay, from one Ti plasmid, one T strand. VD1 and VD2 are involved in the initial T strand processing. So now we have already know about Vir A and G. Now going to talk about the Vir D1 and D2. Vir D1 and D2, they are involved in the initial T strand processing. So T strand processing is done through D1 and D2 and acting as site and stand specific endonuclease. So those are actually, see, in order to make the fragment of tDNA, but first steps would be the cutting of this strand. Now, so endonuclease should be there. So D1 and D2 is playing that role, endonuclease activity. After cleavage, VT2 covalently attaches to the 5' end of the T strand. So in this case, at the 5' end, it is attached at the, actually, I think it is at the right border. At the right border, it is binding with the 5' region here. 5' region, 5' end of the T strand at the right border, nick, and to the 5' end of the remaining bottom strand. So it is uh, after cleavage, D2 covalently attaches to the 5' end of the T strand at the right border nick and to the 5' end of the remaining bottom strand of the Ti plasmid at the left border nick. In this case, left border, right border, both places, D2 is binding by its tyrosine 29. Okay. Now VG nicks the lower strand at the right border sequence and binds to the 5' end. So nicking is happening here. Okay. And one strand only. Okay. And binding at the 5' position. Now, in this case, what is happening then? The T complex is composed of at least three components: T strand DNA molecule and VD2 protein, and around 600 VE2 proteins. So, why those are involved? Because in order to control the exonuclease activity, you have to restrict the exonuclease activity, because after the cutting of one single strand, exonuclease may degrade those part. Now to control that, VE2 is playing the main role here. Okay, 600 number of VE2 is binding at that position. Okay. Now, if VE2 associates with the T strand after intercellular transport, VE1 is probably involved in preventing VE2 T strand binding. Uh, E2 associated with the T strand after intercellular transport. Now, after what happened? Now, single strand is produced. That single strand will be transported to the plant cell. As I told you, na, here single strand is transported here. Okay, so that is single strand is actually bound with the E2 protein, VIR-E2. Now, VIR-E1 is probably involved in preventing VIR-E2 strand binding. Acha. Vir E2 T strand binding. Acha, it is preventing Vir E2 and T strand binding, so that a single strand will be independent. Now, judging from the size of the mature T complex and the inner diameter of T pilus, the T strand is probably associated with Vir E2 after intercellular transport. Acha, after transport also there will be attachment with the E2. Okay. So E1, E2, they D1, D2, those are actually participating in the cutting of the tDNA and transport of the tDNA into the plant cell. Okay. Now, how transportation is happening? Transport of the T complex into the host cell most likely occurs through type 4 secretion system. So, you have to remember that what is the secretion system involved in this process? Type 4 secretion system. Okay, so in the membrane, there will be some system which can transport single stranded tDNA along with VG2. Okay, now, in agrobacterium, the type 4 transporter, which is actually T pilus, it's like pilus. You know, pilus, you know now, bacteria joining. Plasmid can be transferred through the pilus, plasmid
Yeah,
transfer.
like
है ना?
conjugation, no?
Conjugation, yes. So it is like that. Conjugation is happening between bacteria and plant cell. Okay, so this is actually transporter, one type 4 secretion system. So type 4 transporter or T pilus comprises proteins encoded by VIR-D4 and by the 11 open reading frames of the VIR-B opera. So here D4 and VIR-B, those are involved for the type 4 secretion system formation. Okay. Okay. Now, intercellular transport of tDNA is probably energy dependent and requiring ATPase activities from VIR-B4 and VIR-B11. Okay. Now, physical contact between agrobacterium and the plant cell is required to initiate T complex export. So, in this case, obviously, physical contact is there. So, pilus formation will be there. And without recipient plant cells, T strands accumulate when VIR genes are induced. Without recipient plant cells, T stands accumulate. Acha. Whenever plant cells are not there, that means this type of induction is not happening by the phenolics. What will happen? Without recipient plant cells, T strands accumulate when Vir genes are induced. So T strand accumulation will be there. whenever these genes are induced. Okay, induced by the phenolics, but side by side plant cell is not present. So in that case, there will be accumulation of the 10th strand. It will not be transferred to the plant cell na, because side by side plant cell is not present. Okay, so T strand accumulation will be there. Okay, so here you have to remember through which transportation is happening. Type 4 is secretion system, and it is helped by how it is produced, VD4 and VB operon. Okay. Acha. Now you can understand the table. Okay. Again, anything else is given or not? Let's see. Acha. In this case, stages of agrobacterial delivery to plants. Vir A and Vir G system induces TI plasmid. Vir gene expression in response to signal molecules at wound sites. This is the first step. I told you A and G. First of all, A, then G is activated whenever phenolic compound is coming. Now, second step is the VIR-D2. VIR-D2 relaxes and relaxosome components exercise tDNA at ORI-T-like border sequences. So in this case, VD2 is involved for the cutting of that tDNA. Now, third is the recruitment of the relaxase tDNA intermediate to the T4SS involves binding proteins and special determinant. Here C1. Here different way it is written. Different cases, different. But same thing is there. C1 involves binding protein and special determinant VC1. 4 is the T4CP VD4. VD4 is involved. Okay, so again VD2. Okay, now coming VC1, then VD4, which is recognizing protein and nucleoprotein secretion substrates and initiates transfer. So D4 is involved the transfer of the DNA. Now pylene VB2 contacts plant receptor proteins. So that type 4 receptor, that transport system. Now, delivery of bacterial effectors to plant cytosol is followed by VIR-E2, T complex maturation, nuclear import, integration, and transgene expression. Okay. So same thing, but slightly different is given. Name is given differently, nothing else. Okay, so in this case, he pilus, or you can say ki transporter, that is actually vir B2 and B5 here. In previous slide, it is B only. For comprises protein encoded by vir D4, D4 and B. Here it is written B4, D4. Why D is not given here? In different book, you will get different. This is a different problem. है ना, in this slide it is given. See, in agrobacterium the type 4 transporter comprises proteins encoded by VD4 and VBB, but VD is not given here. B2 B5 is given, but D is not given. Why? Don't know. B2 converter receptor, BTI means receptor, pylene with B2. D4 is not given. Why? That is the problem. Okay, anything else given here? First step is the VD2, then with E2 and E1. Acha, with E2, E1, it will attach with the DNA. Okay, and it is going inside the plant cell, and with E2 will be removed. Okay, and what will happen? These are the few proteins are involved for the integration. Okay, rock A, rock 4, site A, these are involved here, and it will be going inside the nucleus, and it will be integrated into the genome. So here neat and clean things are given. See, we D1, D2. Now D2 is attached in order to make the straight line. Okay. uh, Straight line, or what is that, thread line structure. Folding is restricted by this VD2, holding of the DNA. Now this is and with D2, VD2. Oh, red color is for E2, E1. Okay, red D1, D2, VD2 green. Answer here VD2, VD2. This is D2. Distant, then where it should be E2. Everyone is confusing us. Don't see this one. Don't see this one also. Very confusing. You just follow these two PPT. This one and this one. Okay. Simultaneously, it is written properly, neat and clear way it is written. This is not nuclear import of chromatin targeting. Because of the large size of T complex, the nuclear import of T complex requires active nuclear import. T complex nuclear import is presumably mediated by the T complex proteins VD2 E2. Both of them have nuclear localizing activities. Acha. So mainly D2 and E2 are involved. Tata box binding protein, both of which bind to the VD2, and V1 binds to the VD2, which leads to chromatin targeting of T complex. So E2, D2 and E2 are actually helping integration of tDNA into the genomic DNA. Okay. Again, another thing is there. See here, T integration, how it is happening. It's simple way it is written. This is very good slide. See tDNA. See initially at the 5 prime right border. At the right border, D2 is binding. I told you. The 5 prime position right border, D2 will bind. Okay, and E2 is binding on the DNA. Now there will be degradation. So this is actually double stranded, initially double stranded, now it is single stranded. Okay, now what will happen, bacha? DNA polymerase, again double stranded. Now, okay, so now this is actually going to inside the single stranded DNA is actually going inside the plant na.
Mm.
Inside the plant, how it is integrated into the genome, that is given here. First of all, the C is initially it was single stranded. Now few primer will be attached, and DNA polymerase will synthesize the next strand. Okay, and in that case, few other proteins are involved here at this position. Ku80, Ku70, these are attached so that there will be no further degradation of this strand. Okay, and now what will happen? What will happen? The plant genomic DNA, in the plant genomic DNA, anywhere of the plant genomic DNA, this band, the blue color tDNA can bind. Okay, so see, this is plant genomic. Black color is the plant genomic, and middle of the plant genomic, what is present? This is the tDNA. Okay, so in this way, actually simple, if you consider the simple process, so this is happening. D2 and E2. These are majorly involved here. है ना? This is quite good, simple way to understand the process. Okay. Now, look, again, we should go to that table so that if we can understand it or not. Okay. nahi? So let's see what is happening here. So I am deleting everything so that you can understand. Vir A, this is already you know receptor, and it is binding with phenolic compound. Okay, now phosphorylation is happening, and so Vir G protein is also activated by phosphorylation. Okay, now Vir B. Vir B is the membrane protein, possibly form the channel for tDNA. B protein is involved for the type 4 channel, है ना? Through which actually through that channel single stranded DNA is coming out of the bacteria and going towards the plant. So tube formation, conjugal tube pilli formation है ना. Now VC is participating in the it has the helicase activity, binds to the overdriving region outside the right border, involved in unwinding of tDNA. Now with D1 has topoisomerase activity. Okay, so in that case we have discussed now D is attached finally with the single stranded DNA. It has the topoisomerase activity and prevent attack by exonuclease because it is binding at the 5' end of the right border. And VD2 is an endonuclease activity. It nicks the right border. Okay. So exonuclease, endonuclease, both are there. D1 is having the exonuclease activity, whereas D2 is having the endonuclease activity. It has the endonuclease activity. Okay. Now E1, E2. Say protect tDNA against nuclease and target DNA to plant cell. It acts as single stranded binding protein. So this is actually, if you consider vir E, है ना? Vir E, if you consider na, here it is binding at the single stranded na, so
Hmm.
that there will be no bending of the single strand or degradation of the single strand. Okay, so it is acting as the single strand binding protein. Vir E. Now, if you consider about the Vir G, this is DNA binding protein, probably forms dimer after phosphorylation by Vir A and induces the expression of all Vir operons. So initially we have discussed this. A is activated and G is deactivated. And G is actually involved for other. It is acting as the inducer, you can consider. Whenever G is activated, it is actually activating the all other Vir genes. So expression of the all Vir operons. Okay. So main gene you can consider. Vir G is the main. A and G initially. D2 and E2. Now E2, D2 and with E2 have a nuclear localization signal. It has the signal key. You have to transfer from one place to another place. Now with B and with D4, type 4 secretion system. B and D4, these are actually involved in the type 4 secretion system. Okay. B and D4. Remember it. So this table is very important because you will get question from this table. You have to remember. Slightly tricky, but you have to remember. है ना?
Okay, ma'am.
I can understand. It's not easy to
It's
remember.
confusing. All the
Confusing slightly.
things.
But you just remember, understand ki finally what is happening. First of all, tDNA is degraded. That after the degradation, double stranded is not transferred, single stranded one. है ना? In order to protect that single strand, and helical structure is there should not be helical structure. No? It should be like straight line, like chain structure. So in order to maintain the chain structure, few proteins are bound with this DNA. Okay, those are actually with E. And this finally it is transferred to this plant cell through the channel transporter, type 4 channel. Okay, that is, if you don't these things, na, there will be no problem. Initially it was not understood. After the discussion of the process, you understood little more. है ना?
Yes, ma'am.
है ना? So there will be the thing. That is the thing. Okay. Now, with agrobacterial mediated gene transfer. So in this case, what are the requirements? Plants explant mass produce This phenolic compound should be produced. Induced bacteria should give access to cells that are competent for transformation. Then transformation competent cells and tissues should be able to generate into whole plants. Okay. Now what are the advantages? Natural means of transfer, hence plant friendly. It is capable of infecting intact plant cells, capable of transferring large fragments of DNA, and stability of gene transfer is excellent. A very popular system it is. Disadvantage: limited host range, cannot infect cereal plants. Mostly dicot plants are utilized. Monocot cannot be utilized. Okay, dicotyledonous plants are utilized for this purpose. Cereal plants cannot be utilized. Sometimes cells in a tissue that are able to generate are difficult to transform. Now, sometimes cells in a tissue that are able to generate are difficult to transform. Acha. Transformation is slightly difficult. Acha. In this case, TI plasmid based vectors co-integrate and binary vectors. Two type of vector possible. Okay, co-integrate and binary vectors. Now, wild type agrobacterium TI plasmid cannot be used as the gene cloning vector. Okay, so normal wild type agrobacterium TI plasmid cannot be used because of large size. Okay, and presence of oncogenes. है ना, oncogene is present. TI therefore it is tumor inducer. Tumor causing agents, genes, auxin and cytokines are there. Now lack of unique restriction sites and make sites within tDNA. Very less number of restriction sites are there, and there are two genetically engineered TI plasmid. Okay, TI plasmid, but that is not natural one, wild one. It is genetically engineered. Those are actually used. Those are known as co-integrate and binary vector. Okay, co-integrate vector and binary vector. Okay, generally utilized. Uh, i- Now, in this case, making a co-integrate. What is co-integrate vector? So, in this case, what is happening? Both the tDNA with our gene of interest and V region are present in the same vector used for transformation. So, in that case, both the tDNA with, see, it in the normal case, what I told you, this is the TI plasmid. Vir gene should be there for the infection. It is essential. Okay, vir gene, virulence, infection. Now, generally, this tDNA, in the tDNA, auxin, cytokine, those are present. Those are not required. Generally, we delete those cytokine and auxin gene, and we replace with our interest of gene. Okay, so our interest of gene is actually inserted in the tDNA. Okay, along with some marker like antibiotic resistance marker. That is a normal. But here, what is happening? Both the tDNA with our gene of interest and V region are present in the same vector. So in this case, V region is present, tDNA is present, and gene of interest is present. DNA with our gene of interest and V region are present in the same vector used for transformation. At first, an intermediate vector is made. So initially, one intermediate vector is made using E. coli plasmid. Okay, so initially E. coli plasmid is taken. Now, in that case, V region is added. V region is added plus tDNA border added, and then origin of replication added, and this is the plasmid sequence. Okay, so this is E. coli. You can the E. coli plasmid PBR 322. This is PBR 322. Just a minute. What is happening here? You just read it. I'm telling you. This is equal vector.
राती जाना है अभी जॉब दी थी जॉब दी Ma'am, it's muted. Voice.
Okay. So here border regions are there. अरे भैया काहे इतना डिस्टर्ब हो रही है. No, it is not. अच्छा. Okay, just a minute. I'm showing it. First of all, I have to copy this one so that it will be easy to understand. Okay, now this is the actual structure. No? I'm deleting it. Hmm. Okay. So in this case, what is happening? Virg region is there. वीर तो है ही भाई, रखना ही पड़ेगा. वी रीजन. Okay. This is there. TDNA border, that means left border is there, right border is there. So totally this part, if you consider that this part is there, origin of replication is there, this part is also there, and now another sequence is added, that is the plasmid DNA sequence. Okay, TDNA border है, origin of replication है, virulence है, virulence gene है, और बाकी जो plasmid है वो that is from the PBR 322. Okay, so in this case, this part is not present, I think. Auxin cytokine open tDNA is not present. Instead of that, what is present? I think this is actually in this position, there will be PBR, PBR 322. Now, second vector is a disarmed PTI vector. Okay. So in this case, second vector PTI vector plus gene of interest plus some markers plus PBR2, PBR322 sequence. So in the next gene or next vector, what is present? Mainly disarmed PTI vector. That means T is not there. tDNA is not there. Now gene of interest is added. Okay, gene of interest instead of tDNA, what is present? In that case, if you consider this is the gene of interest present instead of tDNA, along with gene of interest, what is added? Along with gene of interest, one marker should be there. And I told you that generally antibody resistant marker is there. So it is there. Okay. Now, if you and some PBR322 sequence. So here it will be like this. Okay. So initially in the first vector tDNA is not present. In the second vector it is actually recombinant one. Okay. Now both intermediate vector and disarmed PTI has some sequences is common. That is the PBR 322. Both of the cases, these sequences are there. Therefore, by homologous recombination, so there will be same sequence for the PBR 322 sequence. So there will be homologous recombination means I told you na homologous sequence should be there for the homologous recombination process. So there will be recombination between these two vectors. Okay, co-integration of two plasmids will take place with agrobacterium. So therefore, by homologous recombination, co-integration of two plasmids will take place with agrobacterium. So in this case, there should be one picture how recombination is happening. Why it is not given, I don't know. Should be given. So there will be recombination between these two plasmids. Okay. So result co-integrate vector that has both tDNA with our gene of interest within the tDNA borders and V regions. This complete vector is used for transformation. One example of this type of co-integrate vector is known as the PGV2260. Okay. So together, after the recombination only, whatever you are obtained, which is known as the co-integrate vectors. So a result is the co-integrate vector that has both tDNA with our gene of interest within the tDNA borders and head regions. Okay. Achha. Just a minute, I had one recombination sequence.
Video is freeze.
Oh, sorry.
The video is not moving.
Hello. Hello. Now is it okay?
Hello, ma'am.
Uh-huh.
No, the video.
Video paused, but
Yeah,
I can see it is working fine. No, it's not paused. Your internet problem is there, I think.
might be the connection then.
Uh-huh. So in this case, co-integrate vector means two vectors are integrated to make one separate vector. Now, if you consider about the binary vector strategy, two vector strategy. So in this case, or these two separate vectors are added. There, two vectors are used based on the knowledge that V region need not be in the same plasmid along with tDNA for tDNA transfer. So in this case, what is happening in one plasmid, V region is present. In another plasmid, V region is not present. Okay, here two vectors are used. Integration, homologous recombination is not happening here. Separate plasmid. Here two vectors are used based on the knowledge that V region need not be in the same plasmid along with tDNA for tDNA transfer. So if V region is, it is present separately. See here in this case V region is there and Ti plasmid without tDNA. If V is present in that case, tDNA is not present.
Ma'am,
And
which slide number are you showing?
21.
I'm still stuck in 20. I'll leave the class and join again. Okay, now it's visible.
Okay.
I was stuck
Uh,
on 20.
it's raining. Okay. So in one plasmid is Ti plasmid without tDNA, but V region is present. Okay. Now in the second one, a recombinant plasmid you can make recombinant plasmid with foreign gene and kanamycin, that is the antibiotic resistance gene, between tDNA and 25 base pair repeats. Okay, so in the tDNA region, your genome interest is inserted and kanamycin gene resistant gene is inserted. Okay, and this 25 base pair repeats are there, left border and right border, there should be there. Okay, and foreign gene kanamycin gene is there, but V region is not present in this case. See, both are different. Now, binary vector consists of pair of plasmids, a disarmed Ti plasmid. This plasmid has tDNA with gene of interest plus origin for both E. coli and Agrobacterium, also called as mini Ti or micro Ti plasmid. like example bean 19. Now, if you consider helper Ti plasmid has virulence region, so in this case, this is the helper one, and this is actually tiny micro Ti plasmid, where your recombinant part is added. Okay, helper Ti plasmid has virulence region that mediates transfer of DNA, tDNA, and micro Ti plasmid to the plant. So, why it is happening? Why this type of construction is done? Because as I told you, normal TI plasmid is longer, है ना? Size is bigger, so it's very difficult to handle. Now separate, if you use it separately, the size is decreased, na plasmid. This only V region is present, है ना? And in this case, V region is not present. But if you add, V region is required for the infection, so if you add it separately, so and two plasmids can be used in a single bacteria. So this is actually done in this way, two vector strategy. This is called the two vector strategy. One vector V region present, another vector T plasmid is present along with E. coli. E. coli is involved for easy to clone and express. So E. coli is given here. Okay. See what is happening if you bacteria invade at wound sites, because wound is giving the signal, Hannah, to transfer the DNA. So in this case, if you cut the leaves piece, okay, and you just put it in the media agar plate with growth hormones and kanamycin. So if kanamycin is given, that means only the recombinant strain will grow, है ना? Because it is attached with the gene of interest, and kanamycin resistant gene is attached with that. So what will happen? They will grow, and these kanamycin resistant plants contain the foreign gene. Okay, in this way it is happening. So initially, in this case, from two plasmids, one co-integrate vectors are produced by homologous recombination. Okay, that is used as the cloning vector. Whereas in this case, two separate vectors are used. In one vector, V region is present. In another vector, recombinant strand. That means foreign gene is present along with antibody resistant gene. Acha. Now, advantages of binary vector over co-integrated vector. Binary vector do not need in vivo recombination. Okay. Recombination, I told na, homologous recombination is not happening in the binary vector, and binary vectors require only an intact plasmid vector to be introduced into the target bacterium, making the process of gene transfer more efficient and quicker. Confirmation of the transformation event is accomplished very easily, because very easy process. Right? Here it's slightly complicated. This process is slightly complicated as compared to this one, two vector strategy. Okay. Now, recent work has been shown that the plant vector KU80 is involved in the tDNA integration process, and most likely by binding between double-stranded tDNA. See, in this case, it is given KU80, KU70. So these are actually recent finding. They are binding with the double-stranded DNA, which is inserted into genomic DNA. है ना? It is there. 80, 70. Those are initially it was not there, but recently it is developed that those are also involved in the uh, in this process. So in addition, overexpression of the East RAD54 protein led to high frequency gene targeting in the transgenic planes. So in this case, RAD54 is also involved. These two reporters further support the notion that integration of tDNA molecules is promoted by host cellular factors. So you just remember from this thing that KU80 and RAD54, those are also involved in this process. Okay. So, with an ever expanding host range that includes many commercially important crops, flowers, trees, species, agrovegetable is guaranteed a place of honor in nearly every plant molecular biology laboratory. Okay. Its recent application to the genetic transformation of non-plant species from yeast to cultivated mushrooms, even human cells, places agrobacterium at the forefront of future biotechnological application. Okay? Nothing else. You don't have to read that. Acha. In this case, this is the expression system. Simple way how it is happening. From the bacteria, DNA is extracted. eh? TI plasmid is extracted. Now cloning is done during the recombinant DNA formation. So in that case, you have to do in this way: cloning and designing the genes. Now transformation into the plant cell and cell and tissue culture, and you can get the plant breed with new expression. है ना? Whatever you have inserted, there will be gene expression for that gene. Okay, understood this thing? How genetic engineering is done. This is the overview.
Yes, ma'am.
Mhm. And here also same thing is given: agrobacterium tumefacciens, ti plasmid, tDNA present, restriction sites are there. Now insertion of gene into plasmid using restriction enzyme and DNA ligase. Okay, so red part is a gene of interest. Okay, so do the restriction and the nucleus treatment for both ti plasmid as well as DNA containing gene for interest. Same restriction enzyme nuclease is given. Now there will be ligation, so we add the ligase enzyme, so you can get the recombinant ti plasmid. Now next step, introduction into plant cells in culture. Okay, you just add this plasmid in the culture. So what will happen, this DNA integration will be there. So tDNA part will be uh, so this part will be, only this part will be transferred, sorry, only this part will be transferred from recombinant ti plasmid to plant cell, and it will be integrated into the genome. Okay, so you will get new expression of that gene. So regeneration of the plant and plant with new trait. Okay, so these are happening actually during the recombinant production. Acha. So TI plasmid vector for creating transgenic plants. So transgenic plants can be created by TI plasmid. Foreign genes could be inserted into the tDNA. So this is actually done during the transgenic plant formation. This is the actual pathway how it is happening. Transgenic plants. Now, foreign genes could be inserted into the tDNA and then transferred to the plant. The modified TI plasmid, the genes responsible for tumor formation are deleted, है ना? Because you are removing cytokine and auxin producing part. So there will be no tumor formation. Only there will be expression of the gene. Now, selectable markers added along with the appropriate regulatory elements. Okay. So if you consider the Kanamashi resistant gene, you can add nothing else. Acha. Here also same thing. How transgenic formation is. This is the normal case. है ना? First one is a normal case. Because in this case, agrobacterium tumefacciens, this is the bacteria inside that TI plasmid is present. It is going inside the plant and it is producing actually crown gall. Because we are not doing any recombination here, we are not deleting the cytokine and auxin here. This is the natural condition. Okay, so crown gland formation. Now if you consider if you want to make the transgenic plant, in that case you have to insert your foreign DNA, whatever you want to express it. So that foreign DNA should be incorporated at the tDNA part, where and mainly at the cytokine auxin part. Okay, and see, this is the recombinant strain, and this recombinant strain is transformed to the plant cell. And what will happen, this part, a single strand is going inside the plant cell, and it will be converted to double stranded DNA, and double stranded DNA will be incorporated into the genomic DNA, genomic DNA of plant. Now you do just and give the kanamycin. Obviously, if you add the kanamycin in the media, only the recombinant strain will grow, non-recombinant strain will not grow. Okay, so you will get the actual desired gene expression in the plant. Okay, so this is called the transgenic plant production. Clear, Beto? Now is it clear?
Yes, ma'am.
Transgenic, how it is forming? I hope it is clear. So we are just inserting the foreign DNA where? At the tDNA part. है ना?
Hmm.
Along with one uh, that gene, what is that called? Uh, Antibody resistant genes. Okay, selector marker, selective marker. Same thing. From several sources I have taken. Same thing. Achha. Is there any limitation? Sure. The production of phytohormones by transformed cells prevents them from being degenerated into mature plants. These are not so important. Based vectors components. The selectable marker genes such as neomycin put under the control of plant transcriptional regulation signal, including both promoter and terminator polyamines. छोड़ो ये सब जानो. This is already we have discussed binary vector system. Okay, this is the binary vector system. See, E. coli origin is there. Okay, and tumefaciens origin is also there. Left border, right border, target gene, plant selectable marker. Okay, gene of interest. Target gene means gene of interest. Now selectable marker gene for both E. coli and tumefaciens. It is also inserted. Co-integrate vector system. The co-integrate vector has a plant selective marker that they target in the right approach. Same thing already discussed, na? This thing I wanted to show you how. See, homologous DNA sequence. I told you, na, in case of co-integrate vector system, by homologous DNA recombination is done. So these two vector systems, they have common sequence for PBR 322. This position, blue position. So there will be homologous recombination in between that. Finally, product will be this one. So in this case, origin is there, bacterial selectable marker gene is there, right border is there, target gene is there, plant selectable marker, homologous DNA sequence. This is the blue one. In this case, recombination is happening. So it will be there. Okay. After the recombination, it will be there. Now left border. This is origin will be there. Virgin is also there. Okay. So this is actually recombinant TI plasmid. Okay. Now tell me, is it possible for you to do? Match the following with proteins with their correct function during agrobacterium mediated transfer of tDNA to plant cells. I told na, say from that table you will get question.
Hmm.
Virgi, nucleus targeting of tDNA, component of membrane structure, transfer apparatus for tDNA transfer, component of membrane structure, proteasome mediated destruction of proteins coating tDNA complex, induction of virgins. Where you are staying? See. See, if you don't know anything, na, if you know only one thing, from that also you can find the answer. See, Virgi, this should be, I think, induction of virgins. Anna, I told you this is the important gene, Virgi.
Hmm.
Anna, first of all, Vir A. VA is actually activator. Sorry, receptor. It is acting as the receptor of what? Those phenolic compound. है ना? A first, it is activating what? Activating VG. है ना? You have to remember this, at least these two things. So A is activating G. Now G is actually activating all other genes. Main purpose of G activation is it is actually inducing other. So only one option is there, A4. है ना?
Second option. Yeah.
हाँ, तो it should be second option, but you can other options you can check. See B1. B1 means D2 is actually it is having the nucleus targeting of tDNA. Nucleus targeting of tDNA. B1. B1 is actually component of membrane structure. Okay. So this is going towards this one. This is this one. And F. So VG पहले दे देते हैं. This one. And this is F. Proteasome mediated destruction of proteins coating tDNA complex. Okay. Proteasome is actually ubiquitin coated proteins destruction is done inside the proteasome. Okay, proteasome mediated destruction of proteins coating tDNA complex. Okay, is it okay, beta?
Okay, ma'am.
Hana, see from the question you have to read full question and answer too. So from the
Yes.
answer you can understand what they are asking. And if you don't know anything, if you know only the one thing, from that you can identify what will be the answer. Now going to next one, this one. So if you understood the process, you should do it. How? What are the sequences? है ना?
Ma'am, the slide is still stuck in 36 for me.
Oh, it's 38. I'm showing 38. It is not visible.
No, ma'am.
Then what can I do? Your internet problem is there, I think. Is it visible? No.
No.
Then wait for 2-3 minutes. Let's see.
Only questions are left, ma'am.
Huh.
This topic.
Questions are left.
Can we end for today then? I'm also not feeling so well.
Okay. We can.
I'll go through the questions.
Yes, you can go through the questions. Is it not visible?
No, ma'am, still 36.
It is
Still
30.
the one with the box.
And now?
No, ma'am.
Okay. Okay. Then what should I do? Bolo. I thought I should teach you another question, another topic. Okay, half an hour is there still. So should I cross this PPT and share
Rose.
another one? No, still it is hanged.
Yeah.
Just I'm changing one. Okay, changing another one. Just a minute. Gene cloning, not gene cloning. Blotting, all blotting techniques. I wanted to explain you that blotting techniques. Let's see, it is visible or not. Blotting technique is visible. No, it's not changed. PPT is changed.
Yes, ma'am.
Blotting techniques available.
Yes.
है ना?
Mm.
Okay. So just shortcut way I'm explaining it. You just listen to me. If same, all the blotting techniques are same. Southern blotting, northern blotting, and western blotting. Okay. Now, before going to the blotting process, you should know cDNA. Already I have discussed. cDNA means complementary DNA, and it is produced from the mRNA by the enzyme reverse transcriptase. Right?
Yes, ma'am.
Huh. So in this process, how cDNA is produced? If you consider you have the mRNA sample, you want to produce the DNA. So in cDNA, in that case, in mRNA, in case of eukaryotic system, poly A tail is present. So against poly A tail, you can make primer. So very simple to make this type of primer because oligo T. You have to add only oligo T. So from that primer, you can make one single stranded complementary strand against mRNA. है ना? First of all, you are making single strand here. cDNA is actually complementary to mRNA, so it is known as the complementary DNA. So it's opposite to mRNA. It is there. So it is single stranded. Okay. Now what will happen? This single stranded cDNA is converted to double stranded by polymerase. Okay. Now double-stranded cDNA ready for insertion into the appropriate folding vector. So whenever you are talking about the mRNA cloning, that means you have to convert it into double-stranded cDNA. Then only that can be inserted into any plasmid. Okay. So this is called the cDNA. Now going to DNA probe. DNA probe means this is actually a short labeled single strand of DNA or RNA used to locate its complementary strand in a quantity of DNA. That is means if you consider this is one single stranded DNA is there, you want to identify that strand. Okay. In order to do that, against this DNA strand, you have complementary strand but not full strand. Okay, only complementary short strand. Along with that, what is present? One marker is present or label is there. Label means either ELISA I discussed now, enzyme was attached with the protein, antibody or antigen. Similar way, here mostly radioactive compounds are attached. Okay, so in this case, short chain attached with the radioactive compound is known as the probe. Whenever this probe is attached with the complementary strand, you can identify this single strand properly. Okay, so in order to identify any DNA or RNA, okay, it is probe is used. So a short labeled means any most of the cases radioactive compounds are attached. Okay, labeled single strand of DNA or RNA used to locate its complementary strand in a quantity of DNA. Okay, now hybridization. This process is known as the hybridization. Here, probe is attached with one strand of the DNA. So here hybridization is happening. Hybrid DNA, double-stranded DNA is produced. So it is known as the hybridization. So in the blotting process, actually we do the hybridization. Okay. Now gel electrophoresis already I have discussed. Gel electrophoresis mainly agarose gel and SDS phase. In agarose gel, DNA and RNA separation done, and in the SDS phase, we generally separate protein molecules. Okay. So this is the gel apparatus. You don't have to know this thing. Now, what is blotting? Blotting means this is actually technique. Okay, these are actually techniques for transferring DNA, RNA, and protein onto a carrier. Carrier mainly membrane is used, so they can be separated and often follows the use of a gel electrophoresis. Now, in this case, before the blotting process, what is done? Gel electrophoresis is done. After their gel, from the gel, proteins or DNA or RNA, those are transferred to any membrane, nitrocellulose membrane, nidone membrane, or other membrane. Okay. Now, separate name is given for the separate molecules. Southern blot is for the DNA transfer. Northern blot for the RNA transfer. Western blot for the protein transfer. Okay. So, in this case, it is written here: Eastern blot, Western blot, Southern blot, Northern blot. Eastern blot means for the detection of protein post-translational modification. So, if there is any post-translational modification, you can detect through the Eastern blot. Now, if you consider the Western blot, we generally detect protein molecule. Southern DNA, Northern RNA. Okay, shortcut way of remembering this thing is that you should remember snowdrop. Snowdrop. Snow means S means southern,
Mm,
it is related to D. Okay, drop
mm,
DNA. And N means northern. This is related to RNA, so R. And O is nothing. And W means western, that is for protein. Okay, so these are not the directions. Okay, southern is actually
mm,
name of one scientist. Okay, and
mm.
this southern blotting is developed first, and related to that all other names are given. Okay, northern, western, those are given. Now now going to the topic, that is southern blotting. If you consider the southern blotting means what I told you, southern means which molecule will separate?
DNA.
DNA. Very good. Now this is the scientist name, Sir Edwin Southern, who developed in 1975 this separation process. Now in this case, what is happened? First of all, you have to do the agar gel electrophoresis, and then you have to transfer
Ma'am,
it to
video is freeze again. It's still in the snowdrop one. 33.
33. It's 10. I'm
Mm,
showing 3. 10. Okay. Still not visible. Then I have to drop
no,
it, or
ma'am.
what can I do? Okay. So then,
I think it's my network.
hmm, maybe. Okay, what can I do? You can leave. Okay. What do you
Yeah, ma'am,
think?
you'll continue teaching or
Huh?
we'll continue tomorrow or you will continue teaching.
What do you want? You tell me. If it is not visible to what to, what is the
Yeah.
necessity of
No, I
that?
mean, ma'am, if I leave, no?
Huh?
Ma'am also will leave, no?
You want to leave? You go. If it is not working, then you can leave. Okay.
Okay, ma'am.
Chalo. Bye.
Okay, bye, ma'am.